Hello students, welcome to the class of Biomolecules and Metabolism. In today's class, we will learn on what is biomolecules, why biomolecules are so important for our body, what are the various types of biomolecules and their properties. Before starting today's class, uh, just have a quick look what we have discussed in our previous class. In previous class, we have discussed on understanding on power of hydrogen, that is pH and buffer, how buffer solution is prepared, what are the importance of buffer solution, uh, why buffer solution is required for some very uh, fruitful and chemical reactions, acid-base balance, process of ionization, why ionization is so important, hydrophobic interactions, what is hydrophobic interactions, what are the factors that play crucial role in hydrophobic interaction like number of carbon atoms, temperature, shape. These are the things what we have discussed in our last class. In today's class, we are going to discuss on what is biomolecules, what are the various types of biomolecules, what are the functional groups that is associated with biomolecules because it plays very important role like different reactions, biochemical reactions. So that is why it's very important to understand the types of functional group and their properties. Importance of buffers in biochemical reactions. So these are the topics that will be covered in today's class. Let's have a very small video clip. All these organisms are made up of biomolecules. Biomolecules, carbon and carbon molecules, they are going to interact with each other and they form molecules. Carbon interacts with hydrogen. So like methane, here carbon, it interacts with four different hydrogen and it creates CH4. So when carbon is present, that compound will be considered as bio or organic molecules or biomolecules, we can say. So here, this is the structure of carbon. Okay, sometimes it reacts with oxygen, sometimes phosphorus, sometimes sulfurs. So carbon interact with different molecules. So here, long chain of carbon interacts with hydrogen. Then this one is again ring shape. Then again this shape, ring shape. Then here the carbon attaches with oxygen. Here carbon is it's a carbon backbone, and carbon is having 4 electrons in its outermost orbit. So you can see here carbon 3 hydrogen. So hydrogen is having 1 electron. So every electron interacts with one electron of carbon. Then here it is functional group OH, functional group. O represents oxygen and H represents hydrogen. So it is called hydroxyl group. So this is one group of functional group you can say, and it attaches with here. So now it is called hydroxyl, and carbon when it attaches with this group, it converted into ethanol. That is called alcoholic group. So this is the property of function. Now different types of another by the same way hydroxy, here you can see carboxyl, sulfhydryl group, sulfur will be present. Amino group, here this is methane, R represents methane methyl group, and amino NH2 phosphate group, here you can see phosphate, phosphorus, attached with 4 oxygen, 1, 2, 3, 4, and this uh, structure represents phosphate group. So carbon backbone, when carbon attaches with functional group COOH, it is called fatty acid biomolecules. When functional group is carboxyl in this carboxyl group, and here amino group NH2 group, so amino acid. So basically 4 classes of biomolecules. One is called protein, another one is called carbohydrate, nucleic acid, and lipid. These are the 4 major classes of biomolecules that we will cover in today. Now, molecules are group of atoms that contains more than one element, and biomolecules involve in maintenance and metabolic processes of living organism. The small reactive groups present in carbohydrates, lipid, proteins, and nucleic acid are known as functional group. What we have discussed in that small video, OH group if attaches, then alcoholic group, that is called alcoholic group, amino acid and H2, COOH, carboxyl group. So different types of small reactive groups when attaches with carbon molecules, that is sometimes carbon attaches and form carbohydrates, sometimes it forms lipid, because you know that in every organic biomolecules, whatever it is, carbohydrate, lipid, protein or nucleic acid, carbon and hydrogen is always there in a specific ratio. So levels of organization here, we have better understanding that the smallest part of anything here, it is called atom. Now when atom combines, then it will create molecule or compound. When it combines, then it forms organelle. When organelle combines, then it creates cells. Similar types of cells when combines, then it creates tissues. Tissue creates organ. Then organ means like liver system, lung system. It is called organ. Kidney. Then organ forms organ system. Okay, and several organ systems when combines, then it creates organism. So this is level of organization that you need to understand. Now types of biomolecules, as what we have discussed in our previous classes, these are the four major types of biomolecules. One is carbohydrate, another one is lipid, next one is proteins, and this one is nucleic acid, that we will look into this class one by one. Now types of functional group. Amino group. Here you can see NS2 is formula. Last you can see it in chain. Here in the chain here you can see NS2. Okay, amino group. In carbonyl group, here its represent formula will be one bond attached with this carbon atom, and this carbon again attaches with one oxygen via this double bond. It is present in aldehydes and ketones. Here you can see when this R is H will be replaced by H, then it will be aldehyde, and when it is R will be present, means methyl group will be present, then it will be considered as ketone. So C double bond O will represent ketone, okay, and this structure represents CHO represents aldehyde. Okay, so when here this H will be replaced by R group, then it will be considered as a ketone. Carboxyl COOH group, so organic acid, present in organic acid. Here you can see COOH. One O is attached with this carbon and one OH hydroxyl group is attached with this carbon. So this collectively called carboxyl group. Then hydroxyl OH alcoholic group. Here you can see OH. Okay, so this is called alcoholic group. And sulfhydryl represents SH. Many cellular molecules it is present. Here it is in cysteine. You can see SH group, sulfur and hydrogen is attached with this carbon atom. So this creates cysteine. Okay, so this is various types of formula. Okay, and that is present in these categories, and these are examples. Characteristic features of functional groups. Most functional groups are polar. Okay, because in like oxygen and hydrogen, they are having different electronegativity. Okay, so this is why most functional groups are polar in nature. Most functional groups are ionic. Ionic means a transfer of electron. Okay, so that is why it is called ionic in nature. Functional groups are soluble in water due to their polarity, of course. Functional groups have higher forces of attractions. The non-polar portion of biomolecules is chemically unreactive. That is unreactive. Long polar portion of biomolecules means long chain of carbon. You can see that is unreactive, that will be remain unchanged. But the smaller group, that is functional group, you can see that involves or plays very important role in any biochemical reactions. Some functional groups act as acids by donating a proton. Example, carboxyl group COOH can release a proton to become COO minus. Some functional groups act as bases by attracting a proton, that is amino group NH2. Here you can see can attract a H plus ion to become NH plus, NH3 plus. So types of biochemical reaction. All biochemical reactions involve breaking or making. This one is important to remember. Breaking or making of chemical bonds. Thousands of biochemical reactions occur inside a cell. In every second, almost 5,000 of reactions occurs in a single cell. There are 6 major types of reactions that are common in biological process. One is condensation or dehydration reaction. The removal of hydrogen and OH group from two different reactants to make a water molecule is known as condensation or dehydration. This reaction is commonly used to join a smaller molecule into large macromolecules. Here you can see this one is one glucose molecule and this one is another glucose molecule. Both these glucose molecules reacts with each other. Here one water molecule will be released and it converted into maltose. Okay, so this is monosaccharide. Now it converted into disaccharide. So this process is called condensation or dehydration. Dehydration, why it is called dehydration? Because here one molecule of water is released, and condensation, a smaller one converted into complex one. Okay, two molecules here. Then next one is hydrolysis. In hydrolysis reaction, water acts as a reactant to split a larger molecule into smaller molecules. The hydrogen and hydroxyl groups, OH groups, are attached to two products that are formed after reactions. Here you can see this one is a structure of maltose. One molecule of water is going to be added. Here you can see this one is one, and this one is another one. So because of this hydrolysis, complex structure of this molecule converted into simpler one. Maltose is converted into two molecules of glucose when we added water molecule. Then third one is neutralization. When a reaction involves an acid and base to produce salt and water is called as neutralization. Example is HCl plus NaOH. It converted into NaCl, here Na and this Cl. Okay, so NaCl plus again one molecule of water will be released. Oxidation, loss of one or more electrons from atom or molecule is known as oxidation, means loss of electrons. Simply you need to remember here NADH, but after the reaction it will be NAD plus and removal of one electron. So this process is called oxidation. Just opposite to oxidation is called reduction. Gain of one or more electrons by an atom or molecule is known as reduction. Here you can see NAD plus. Then one electron is going to be added, and after this reaction you can see NADH. So this is called reduction process, just opposite to oxidation. Then one is called redox reaction, when both oxidation and reduction takes place. That kind of reaction is called redox reaction. A chemical reaction where electrons lost by one atom are gained by another atom. So here you can see chlorine is having 0, means in this situation, in the current situation, chlorine, whatever the electron is there, chlorine is not going to donate or accept. And here it is sodium and iodine, NaI. So this is another one, and here chlorine, this one. After this reaction, you can see sodium reacts with chlorine. So sodium reacts with chlorine, okay, and iodine will be released. So here the oxidation state you can see 0, but here it's -1. It receives 1 electron, okay, but here sodium is having +1, and here also sodium is having +1. So nothing is changed with sodium. But in iodine, here you can see iodine is having -1, but here in free stage you can see iodine is having 0 stage. So this one is oxidation, and for this 0 to -1 is reduction. So this is called redox reaction, where both oxidation and reduction take place. Now, buffers for biochemical reactions. Buffers often are overlooked and taken for granted by laboratory scientists until the day comes when a bizarre artifact is observed and its origin is traced to a bad buffer. Although mistakes in the composition of buffers have led occasionally discoveries such as the correct number of human chromosomes, okay, using the proper buffer correctly prepared can be key to success in the laboratory. How buffer works. Exclusion by biological membranes. This is not important for all biochemical reactions. However, if this is an important criteria for your particular experiment, it is helpful to remember that deuterionic deuterionic buffers, positive and negative charges on different atoms within the molecules, do not pass through biological membranes. Examples of deuterionic buffers include MOPS and HEPAS. Trace and phosphate buffers do not isomerize into deuter ions. Minimal salt effect. In other words, the buffer components should not interact or affect ions involved in the biochemical reactions being explored. Buffers for biochemical reactions. Minimal effect on the dissociation from changes in temperature and concentration. Usually there is some change in the dissociation with a change in concentration. If this change is small, stock solutions usually can be diluted without changing the pH. However, with some buffers, changes in concentration have more effect on dissociation, and a stock solution cannot be diluted without significantly affecting power of hydrogen. For instance, the power of hydrogen of trace decreases approximately 0.1 pH unit per tenfold dilution, and the pH could change dramatically if you dilute a working solution and at the limits of the optimal buffering range of the trace. Optimal buffering range pH 7.3 to 9.3 at 20 degrees Celsius. Note that trace is not one of goods buffers. A pKa between 6 and 8, most biochemical experiments have an optimal pH in the range of 6 to 8. The optimal buffering range for a buffer is the dissociation constant of the weak acid component of the pKa, buffer pKa plus or minus pH unit. Solubility in water. Biological reactions for the most part occur in aqueous environment, and the buffer should be water soluble for this reason. Prepare buffer at appropriate temperature and concentration. Because changes in temperature can be associated with a shift in dissociation, prepare your buffers at the temperature at which you will be performing your experiments. If your experiment involves a change in temperature, choose a buffer with a pKa that accommodates it. Changes in concentration also can be associated with a shift in dissociation. So if you plan to maintain buffer stock solutions, make sure that the pH adjustment is made after you have diluted the stock to the desired concentration and equilibrated it at the appropriate temperature. Or at the very least, check the pH after dilution. Now, these are the summary of lecture, what we have discussed in today's class. In today's class, we have discussed on what are biomolecules. We have discussed different types of functional group, their properties, their importance, types of biochemicals we have discussed, importance of buffers in biochemical reactions. We have discussed in today's class. In next lecture, we will discuss on what is macromolecules, why macromolecules are important for our body, for living organisms, various categories of macromolecules. So these part we will cover in next class. Now, coming for the questions for practice. Now, questions are these are for just you can prepare your answers for these questions. Give examples of how the study of living materials requires an understanding of chemistry. Describe the various types of biomolecules like carbohydrate, protein, fat, nucleic acid. You can mention. You can also mention their important role. Describe the types of functional groups, what we have discussed in detail in today's class, amino group, carboxyl group, alcoholic group. So you can mention that functional groups, their properties, their reactions, important reactions you can mention or you can remember. Explain why buffers are so important in biochemical reactions. As you know, buffers, if you in buffer solution, if you are going to add a small amount of acid or base, even then that solution will be neutral. Neutral means if that solution is acidic, then remain that solution will be acidic after adding acid, a small amount of acid or base. This is the one of the most or characteristic feature of buffer solution. So these are the things that you need to remember for your better understanding. And that's it for today's class. Till then, bye bye. These are the references if you want to go into detail what we have discussed in today's class.